So one way through which you can do this very elegantly and in a very precise manner is by use a technique which is called RNA interference. The way this works is in a very basic way depicted in that cartoon. Basically, this is a way through which you can target at will the machinery responsible for the production of your protein of interest, and it's working exactly like it's shown there, like a scissor. So you have a way to direct the cellular machinery to cause the degradation of only the information required for the production of the particular protein you care about, and totally ablate the production of that protein from the cells. There's this way now that scientists have developed to do this in a very efficient and specific way. Actually, the work that led to this discovery was acknowledged with the Nobel Prize in 2006 with a scientist that was actually working in the same institute where I was doing my postdoc in US. And so we decided to test this possible approach also in the case of FSHD. And what we decided to test, of course, was, since, again, the only mouse model that is available is the FRG1 mice, we decided to test for FRG1. And basically what we decided to do was to use, so now, as I said, there's a way in which in the lab you can target specifically the information required to make that particular protein and prevent the production of that protein. But the next step, especially in the case of a disease like muscular dystrophy, is that you need to be able to veiculate that machinery to block protein production to the skeletal muscle. You need an efficient way to reach the skeletal muscle. You need an efficient way to reach all the different muscle of the organism, because in the case of FSHD and other muscular dystrophy, you have several different muscle types that are affected by the disease, and so you need to be able, with the less invasive treatment as possible, to reach all the different muscle groups. One way you can do this very efficiently is by use viruses. So there are viruses that have developed through years of evolution a very sophisticated way to go and infect different tissues. In particular, there are particular viruses that are very good in reaching out the skeletal muscle. This virus that we happen to choose is called Adeno-Associated Viruses, or AAV. This is a virus that is several people in the gene therapy field are using to test, develop potential therapeutic approaches for several genetic diseases, and is for two main reasons. One, that is that, as I said, is very efficient in reaching the tissue of interest. And the second is that it's a very safe virus, because this is a virus that is present, most of us have encountered in our life, and is not associated with any pathology. So it has a safety profile much better than other viruses that other people are considering for gene therapy approaches. And so the way what we decided to do was to develop these molecules targeting FRG1. We put them inside this AAV virus, and we deliver those molecules in mice. So again, when I was a postdoc, I developed these transgenic mice overexpressing FRG1. They have a muscular dystrophy. What we did was to treat animals after the onset of the disease, because we wanted to be, in a way, close to a possible future therapeutic approach, whereby most likely we will have to treat individuals after they have developed a sign of the disease. So we did the same in the mice. We treat animals that had already developed muscular dystrophy. We did a single treatment. So the way it's done in mouse is that you do a single injection in the tail, and the virus spread out in all the different muscles of the animal. It's a very efficient way of delivering the therapeutic molecule that you are investigating. And what we did was then, after a long time after the treatment, because we wanted to make sure that the treatment was not only effective but lasted a long time, we evaluated a number of parameters to investigate how good was this treatment in ameliorating the disease in the mouse. And we show you just a couple of examples of what we have done, because this has been published already last year. So one of the things that you can do is to look at the way muscle look. You do that by taking pieces of muscles from the animal, and you cut them and you stain with specific dyes that allow you to highlight the structure of the muscle cells, and then you look at these under the microscope, taking pictures like in this case, and you look at the morphology. So the way a normal, healthy muscle should look is that you should have muscle fibers more or less all of the same size, with their nuclei on the periphery, and just about it. So you should see basically just muscle cells over there. In a muscular dystrophy muscle, instead, you see a quite different picture, which is actually what you see on the top there. This is an example of the muscles of the FRG1 mice treated with a control molecule that is not supposed to have an effect in ameliorating the disease. And indeed, the muscles show all the typical features that you see in a muscle affected by muscular dystrophy. So you see infiltration from immune cells, so inflammation in these muscles. You see muscle fibers of different shape. You see sign of what is called necrosis, so death of the muscle fiber, which is then substituted by new fibers through regeneration process, and so on and so forth. At the bottom, instead, is an example of the result we obtained by, as I said, a single injection with this virus, where, as you can see, the aspect of the muscle, although it is not normal, it is quite different, quite better than the muscle treated with the control, the placebo, let's say.
Can I just ask a quick question on that? Were those pictures that you showed, as it were, universal in the respective mice, or were there patchy changes in the diseased muscle and patchy changes in the treated muscle, or not, or were they, or did all the muscles in the mice, fore them, behind them, and axial muscles look like that on top, and then the untreated ones look like that on the bottom? So
in the untreated animal, yes, there is a little bit of variability from animal to animal. We didn't investigate that too much, although could be interesting because there is variability.
Muscle within the one animal is really what
well, muscle to muscle within one animal, yes, because one of the interesting features displayed by the FRG1 mice is that there is, as you know, so in the disease there is variability in the extent of the disease in the different muscles. The FRG1 mice display exactly the same distribution of muscle weakness that has been reported in FSHD patients. So exactly the same muscles that are affected in the patients are affected in the mice. The ones that are not affected in the patient are not affected in the mice. Very simple example. So the diaphragm, which is never affected in FSHD patients and is never affected in the FRG1 mice. So this is work that has been done when I still was in the US. We compared 13 different muscle groups from the mice, and there is, except for one muscle, there is a complete correlation between the patients and the FRG1 mice in terms of the fact that the muscle is affected or non-affected and the degree to which it is affected in the patients and in the FRG1 mice. No, this is not present. Right.
You didn't see that in the
This is not present. So the asymmetry that is present frequently in FSHD patients in the FRG1 mice is not present. We think this is probably due to the fact that these have to do with the fact that there is an epigenetic component in defining FSHD, which is probably driving differential expression of TAX4, FRG1, you name it, differently in different muscle groups or different side of the body of the patients, and we are not modeling these in the mice. So in this particular mouse, we are modeling only the final stage of the disease, that is simply overexpression of a candidate gene. You know that now we are building a new mouse model where we hope to be able to model also these interesting aspect of the disease, and I will briefly mention about it at the end of my presentation.